Hello and welcome to IB Biology. This is a continuation of Biology Topic 1.1, which is around the topic of cell theory. Today we're going to be looking at the difference between resolution and magnification. Now it's really important that you understand these key terms. Resolution refers to the clarity of an image between two specific points. Now, one way to think about it would be if you were to buy a high resolution television, normally costs more money because the picture quality is very good, i.e. it is able to distinguish between two clear points of an image. If we think about the word magnification, you would use a microscope to magnify an image or make an image appear larger. So keep those two ideas in mind as we go through this section, and we will be looking at the electron microscope and the light microscope in a little bit more detail. So in this case you can see from the image of a cell that we have here that there is a nucleus, a nuclear membrane, we have a cell membrane, we have ribosomes, there are some mitochondria, and these images can be taken from both a light microscope and an electron microscope. So now, as much as this is a very brief overview, it is not completely exhaustive. There are a few main differences that you should be aware of, i.e. the light microscope. You may normally have these in a laboratory, in your school or institution, where you're able to pick them up, you're able to move around with them in the lab, you can take them outside, and they're very portable, they're light. They're relatively inexpensive, they're quite cheap in comparison to an electron microscope. Specimens used can be both alive and or dead, and the actual preparation time for a specimen on a slide is very short. For example, if you wanted to look at some stomata and guard cells on the underside of a leaf, you could do that by using some nail varnish, letting it dry, get some sellotape, use the sellotape to peel off the nail varnish, and then use the sellotape as your slide, which can go on the stage underneath the lens, and then you can look at your cells. So another key difference is the magnification. A light microscope can magnify normally around to 2,000 times magnification in comparison to electron microscopes, which can magnify over 500,000 times. It is worth noting as well that some electron microscopes can magnify up to 2 million times. As we mentioned previously, they are very expensive. And another key difference is that the specimen must be dead in order for it to be viewed with the electron microscope. There are two types of electron microscope that you need to be aware of. One is the transmission electron microscope, or TEM, which can be used to view very thin sections of a specimen. And the other one is the SEM, or scanning electron microscope, which will produce an image of surface structures only. So as you can see from the diagram underneath, okay, we have a picture almost upside down in a way really of the light microscope, where at the top we have the light which shines through a condenser, then it goes through your specimen, the objective lens, and then it's projected through to your eye. In comparison to the electron microscope, which fires quite literally electrons through a condenser, which goes through the objective lens, then onto the specimen before it is picked up by an electron detector. So it's a very, very different way in which samples are viewed using magnification or microscopes. Please do like and subscribe to this video. If you have any comments or questions, please do feel free to post them below, and I will do my best to get back to you as soon as I can. Thanks for watching.